This video will describe the research on how maternal style selectively shapes amygdalar development and social behavior in rats genetically prone to high anxiety, originally conducted by Cohen et al. It has been shown that children who are raised by depressed mothers are more prone to developing psychological problems as opposed to children raised by mothers who are not depressed. The likelihood that children raised by depressed mothers will develop psychological problems can be decreased by enrolling these children in childcare starting at an early age. Previous experiments have shown similar findings as these in rodent models. This experiment dealt with Sprague Dawley rats who were bred to be either high responder (BHR) or low responder (BLR). High responder rats exhibited characteristics such as vigorous exploration of novel or new environments, impulsivity, and were prone to addictive behavior. Low responder rats, on the other hand, exhibited behavior consistent with anxiety and depression. The cross-fostering paradigm was used in this experiment in order to see the effects of different maternal styles. In the cross-fostering paradigm, male low-responder offspring were randomly placed in one of three maternal situations. The first allowed the offspring to be reared by their biological low-responder mother. The second placed the offspring in the care of a foster low-responder mother, and the third situation cross-fostered the low-responder offspring to a high-responder mother. The control in this experiment was a high-responder offspring being reared by their biological high-responder mother. This is able to be the control because previous studies have shown that high-responder offspring are resistant to the effects of cross-fostering. The first experiment simply tested for the impact of cross-fostering on anxiety, depression-like behavior, and social behavior of adult low-responder males. The second experiment looked at the changes in the amygdala and hippocampus of the developing brain. The purpose of this particular experiment is threefold. First, we wanted to examine the contribution of maternal behavior on brain development and behavior in the high anxiety, depression-prone, low-responder offspring. Second, we wanted to illustrate the impact of distinct maternal styles on the adult brain and behavior. Third, we wanted to track developmental changes in the limbic brain when the early life experience is ongoing. First and foremost, all experiments were conducted in accordance with proper animal care. As was mentioned before, the first experiment tested the offspring's behavior. Behaviors were measured in 5 different scenarios. The first scenario was the open field, which was a black box as seen here. The rat was placed in the corner of the box and allowed to freely explore for 5 minutes. Behaviors that were tested included latency to enter the center of the open field, the amount of time the rat stayed in the corner, sides, or center of the open field, and the distance the rat traveled in each of these areas in the open field. An observer blind to the experiment recorded grooming, rearing, and stress-induced defecation. The second scenario was the elevated plus maze, as seen in the lower right corner. The rat was placed in the center of the maze, facing a closed arm, and allowed to explore freely for 5 minutes. Behaviors that were tested include latency to enter the open arms, the amount of time spent in the open arms, the closed arms, or the center square, and the total distance traveled over the full 5 minutes. An observer blind to the experiment recorded stress-induced defecation. The third scenario was the novelty suppressed feeding. This test was conducted in the same box as the open field test. The rats fasted overnight prior to testing. The food pellet was placed in the center of the open field, and the rat was placed in the corner. The rat was then allowed to explore freely for 10 minutes. The behaviors measured in this experiment included latency to touch, sniff, or eat the food pellet. The fourth scenario was a 3-day social interaction test. Each day, the rat was placed in a clear chamber containing a smaller interaction box as seen on the left, and allowed to explore freely for 10 minutes. On the first day, the smaller interaction box was empty. On the second day, it was occupied by a novel male rat, and on the third day, it was occupied by a novel female rat. The novel rats were already present before the experimental rat was placed in the chamber. An observer blind to the experiment recorded grooming behavior and stress-induced defecation of the experimental rat. The fifth and final scenario was a forced swim test. One rat was placed in a clear cylinder filled with 30-centimeter deep water, similar to the picture on the right. The first day was considered a pre-test phase in which the rat was in the cylinder for 15 minutes. On the second day, the test day, the rat was left in the cylinder for 5 minutes while an observer, blind to the experiment, assessed how much time the rat was immobile or simply floating in the water. As mentioned before, the second experiment in this study tracked the changes in the amygdala and the hippocampus of the developing brain. In order to track these changes, brain tissue from the amygdala and the hippocampus was taken from rats at P7, P14, P21, and P75. A microarray analysis was performed following experimental grouping. The grouping was low responder, high responder cross-foster versus a low responder control, and a low responder foster versus a low responder control. The analysis allowed us to see the expression or lack of expression of particular genes in the amygdala and the hippocampus and compare the results to a control. Quantitative real-time PCR verified the particular genes.
BHR rats displaying a greater risk-taking, impulsivity, and aggression relative to the BLR rats, which showed high levels of anxiety/depression and certain risk vulnerabilities. The BLR-BHR cross-fostering offspring spent similar low amounts of time in the center as BLR controls in the BLR fostered rats. The BHR controlled spent more time in the center than all BLR controls. Cross-fostering BLR rats didn't have an effect on how much time that was spent in the open arms. All BLR rats had spent little time in the open arms. Cross-fostering BLR offspring exhibited less grooming compared to the BLR controls, indicating less anxiety. The BLRs groomed less than the BLR controls and the BLR fostering offspring, but did not differ from the cross-fostering BLRs. Cross-fostering BLR showed less stress indicated defections in the EPM relative to the BLR control, another indication of reduced anxiety. BHR controlled showed less defections compared to the BLR controls and the BLR fostered rats, but did not differ from the BLR offspring cross-fostered by BHR mothers. In the OF test, the BHR controlled showed greater overall activity compared to the three BLR groups. Although BLR offspring cross-fostered to BLR mothers displayed slightly but a significant increase in naval induction activity compared to the BLR control raised by their biological BLR mothers. In the EPM test, the BHR controls again showed greater overall activity to the three BLR groups. Maternal care conditions did not affect the total activity of the BLR offspring in the EPM. In a social interaction test, BHR controlled showed much higher levels of interaction with the naval object. A male stimulus rat and a female stimulus rat compared to BLR controlled and BLR fostered offsprings indicated by symbols A or C. Cross-fostering significantly enhanced the social interaction of BLRs, with BHR fostered group showed more interaction compared to the two other BLR groups indicated by symbol B versus C. The BHR controlled showed much less stress-indicated defection in the social interaction test compared to the BLR groups indicated by symbol A versus symbol B and C on Figure 2. Cross-fostering BLRs displayed less stress-indicated defections, indicated less anxiety during this test relative to BLR control. In the NSF test, the BLR showed shorter latency to approach the food zone compared to all three BLR groups. Cross-fostering did not impact this measurement in all three BLR groups, showing similarly high latencies. BLR controls spent more time grooming during the NSF test than all overall groups. This is a general effect of fostering on this measurement, as both BLR fostering and BHR fostering groups showed significantly less grooming in the NSF compared to BLR controlled. In the FST test, BHR controlled showed low levels of not moving compared to the three BLR groups. There was no effect on maternal care conditions on FST movement of the BLR offspring. Microarray analysis revealed robust development gene expression change in the amygdala, but not the hippocampus, of BLR offspring that were raised by BHR fostered mothers compared to BLR controls. There were minimal gene expression changes in the amygdala and the hippocampus of BLR fostered offspring. Figure 2 shows the number of genes significantly upregulated or downregulated in the amygdala. Cross-fostering has little effect on BLR amygdala gene expression at P7 and P14. Numerous gene changes appear at P21 and persist into adulthood, with the majority of alternate genes being upregulated in the BLR-BHR cross-fostering offspring compared to the BLR control. Critiques of this experiment would be that they only used male rats, the scenarios were done at different time lengths, and that behavior is a subjective thing for a person, so it would be hard to quantify the results. For future research, they should try and use female rats to test if cross-fostering has the same effect as it did for the male rats. Also testing to see if the offspring of the cross-fostering BLR have kept the changes in the amygdala that help decrease anxiety and depression.